Hello, good morning. My name is Rene, and today I'm going to present you the article titled An Efficient Protocol Towards Site-Specific Clickable Nanobodies in High Yield Cytoplasmic Expression in Escherichia coli combined with integrin-mediated protein ligation. So, as an introduction, we should talk about what is bioorthogonal chemistry, since the authors of this article focus a lot on using this kind of chemistry to modify the nanobodies. So bioorthogonal chemistry is a set of reactions that can take place in biological environments without affecting biomolecules or interfering with biochemical processes. And in order for biochemical reactions to be considered orthogonal, they must occur at temperatures and pH of physiological environments, and the reaction should involve functional groups that are not naturally present in biological systems. So here you can see some of the groups that are generally used in bioorthogonal chemistry, and as you can see, they have mostly double and triple bonds. So all of these groups have been shown to be able to allow site-specific protein modification. So nanobodies are the heavy chain part of antibodies that come from camelids, and they are quite new and quite useful since they are smaller than regular antibodies. So they allowed a more specific and efficient targeting towards proteins for therapeutic and biosensing applications. So that's why they are said to be tiny but mighty. So as I said, they are used for targeting and biosensing. They are easily engineered, meaning that we can modify the plasmid in order for them to be engineered however we want. And they can be functionally expressed in E. coli, which is a very easy to use bacteria. So in the sense of using them for targeting and biosensing, we can modify them by making modifications in the C-terminal. So since there are protein, there's an N-terminal and a C-terminal, so the modification is often made on the C-terminal since it doesn't affect the binding capacity of the nanobody to the target protein. So one of these modifications techniques is called integrin-mediated protein ligation, which we can see in this image, which involves first having our protein fused to an integrin and that ketene binding domain, which allows the adherence of this whole construct into a column for it to be purified. And then there occurs this NS shift, which you can see here that the NS kind of switch places. And then there can occur a native chemical ligation with any nucleophiles. So in this case, it can be with DDT or an cysteine alkyne, which the cysteine alkyne is actually the purpose of this paper. So native chemical ligation is explained here in further detail, where you can see that two proteins bind together when the sulfur attacks the carbon. And after that, there occurs a shift, and we get this rearrangement of the peptide. So in this case, now that we had the native chemical ligation where we attach this DTT to our protein of interest or the cysteine alkyne to the protein, we do mediated protein purification to obtain our final protein, which is possible because the integrin is self-clippable. So the CBD attaches, as I said, to a ketene column, and the protein can elute when adding specific buffers. And so the cleavage is induced by a strong nucleophile. And as a final result, we have an alkylated protein, which this alkyne is available for the click chemistry, which is explained in this image, where an acid containing molecule is united with an alkyne to give this triazole, which is useful for the binding of the nanobodies to target proteins. So in this paper, the target protein is a VCAM1, which is important because it mediates the addition of to vascular endothelium of lymphocytes, monocytes, eosinophiles, and basophils, and it functions in leukocyte endothelial cells, developing a role in atherosclerosis and being a potential marker for types of cancer. So it as a target protein is very useful to have an antibody that can bind to it for diagnostic and therapeutics. So this article aims at developing an efficient protocol for engineering and expression of NPCAM1, since the previous protocols involving IPL and click chemistry show very low yield of the expression of the nanobodies. And this expression is intended to be with intein CBD and, as I said, subjected to IPL-mediated alkylation. And they'll be doing this by comparing different conditions. The gene has been modified to be expressed in the periplasm or in the cytoplasm of the cells, of the E. coli cells. Also, the C-terminus of our protein can have an intein aplase peptide, an intein lay peptide, or a histactine peptide, which vary in size, and this allows for better cleavage of the protein. Then they also compare the expression in 2 E. coli strains and in 3 media, which is LB, TB, and Preso. So first, they designed the nanobodies by engineering them. So here you can see that the first 3 nanobodies are meant to be for periplasmic expression. And in this case, these 2 are used as a control, while this one, they add the intein CBD. Then the last 4 nanobodies, they're meant for cytoplasmic expression. And here is like the control nanobody for cytoplasmic expression. And in these last 3 is the different spacer that they add, his 6 tag, the aplay, and the lay. And yeah. So to obtain an optimization of the protein expression, they first compare the expression between construct 3 and 4, which means that they compare the expression between periplasm and cytoplasmic expression. And they do this by transforming the plasmids into the strains, and then they transfer it to a pre-culture and eventually to a main culture. So they can be expressed by changing the concentration of IPTG, which is the inducer that allows the protein expression at different concentrations. They also let these cultures grow at different times and at different temperatures. And then they proceed to harvest and analyze the obtained samples in SDS page, and then scale up with the best conditions. And once they have the best conditions, they compare the spacer peptides on IPL mediation, alkylation efficiency, and they do this by selecting these conditions for periplasmic expression. So they extract the nanobody, and after they have stabilized it, they do the SDS page, and the same for the cytoplasmic expression, but the temperature and time were different for this expression. Then they proceed with the IPL mediated alkylation and the cleavage of the nanobody in the column by using these buffers with these 2 alternatives. Either the buffer has DTT or the buffer has the cysteine alkyne, which is the main interest, but they also add mesna and TCP, which are meant to allow a better modification of the C-terminal. Then they do the ZS page of the eluates and dialysis of the nanobodies for them to be more pure. And they do the same experiment, but by changing the E. coli strain and also the media. So they also do this in Empresso and TB. As for the characterization of the nanobody, they do electrospray ionization for transfer mass spectroscopy to confirm masses and enzyme-linked immunosorbent assay to determine antigen binding capacity and the click reaction with an acetobiotin derivative to label the molecules that have been functionalized with alkyne groups and confirm the clickability so they can be actually used for targeting purposes. So as the first result, what we can see is that the most intense bands, they say to be these ones, which is the one with an IPTG concentration of 0.5 millimolar at 18 degrees Celsius, and also the same concentration of IPTG, but at 37 degrees Celsius for the cytoplasmic expression. So these ones were the ones that they used for further expression tests. Then as for the spacer cleaving efficiency, what you can see here is the bands corresponding to the protein uh, uh, unified with the spacer. Here is the protein alone, and here is the sorry, this is the intine and this is the protein with the alkyne. So if you don't see the protein with the alkyne, it means that it was I mean, yeah, that it eluted completely. So from here we can conclude that there was an 100% efficiency with all three spacers. Which we can see in this part and in this part, meaning that all three spacers can be attached to the nanobody. Also, in terms of comparison of yields of the nanobodies with different spacers and expressing different strains in culture media, what we see here is that we obtain a higher expression with the lay spacer and also with the shuffle T7 strain, as you can see here, for all nanobodies. And they chose to do the different media with the lay spacer, but so as you can see, the highest yield is with the Empresso media, but in terms of cost and time, the authors conclude that it's better to use the LB because it's cheaper and faster than Empresso, even though it has a lower yield. So as for the results of the ELISA test, we can see here that all nanobodies, independently of the construct, they have almost the same binding capacity, which tells us that they are all active for targeting the VCAM1. And here what you can see is that this band indicates the clickability of our nanobody to the acetobiotin. So in this case, the most clear band and with most pure is the lay band. So that's why they chose to use this spacer for future engineering of the nanobody. Then for the ESI FTMS spectrum of the lay spacer, what we can see is that after purification, Using this buffer, the N2 aerated PBS buffer, it avoids oxidative couplings, mesna conjugation, and dimerization, since we can see an only peak, which tells us that it's very pure. And in case of this graph, what we can see is that in comparison of the nanobody alone with the alkyne, we can see a 40% conjugation of this nanobody with the biotin acid, which tells us that it's pretty efficient for targeting. So they chose the lay peptide because it causes less mutations and higher alternation degrees. So as a summary, the VCAM targeting nanobody was engineered towards the site-specific incorporation of a bioorthogonal alkyne function at its C-terminus via the IPL technique. And the most efficient nanobody was the one with the lay spacer through cytoplasmic expression with the E. coli shuffle T7 strain at 37 degrees during 3 hours in LP medium, having a final yield of 22 milligrams per liter of culture and 100% of alkylation efficiency. And also by the click chemistry, we can say that the nanobody is successful in preserving the antigen binding. Well, this is by ELISA actually, preserving the antigen binding capacity as the unmodified nanobody. And now by the click reaction, we can see that the coupling, the nanobody is successful in coupling to an acetobiotin derivative using copper UACC click chemistry. And that's all. Thank you for your time.